In this lecture, we're going to look at some optimization methods used in bioinformatics, in addition to what we saw before with dynamic programming. So we're going to look at gradient descent, which can be used when we have a target function that has a continuous derivative, for example, a quadratic error function, and we're going to look at that example to fit parameters to a model using the SciPy library. We're going to also look at Monte Carlo methods, which depend on random sampling to give us either a way to find modifications to a solution that will improve it, or especially allows us to sample many different possibilities and then even obtain statistics and so on. And we're going to see more of that on the second lecture today. And also we're going to look at evolutionary computation, which is inspired in biology and in evolution and uses populations of solutions to try to improve some results, some measure of performance, of fitness, and thus obtain good final solutions for some problem. We're going to focus on genetic algorithms, which is a family within evolutionary computation, and we're going to look at an example for multiple sequence alignment. So let's start with what we saw so far. There are cases where we can find a practical algorithm that guarantees we find the optimum solution for some problem. And we saw this with dynamic programming by taking advantage of the structure of the problem, which allows us to break down into sub-problems and find the optimum for the large problem by finding the optimum for each sub-problems. So with that, we solved the problem of aligning two sequences with dynamic programming. Those algorithms that we saw, whether for global alignment or local alignment, guarantee that we find an optimal solution. That is, one solution for which there is no better alternative. There may be several solutions that are identical, but there will be no better alternative to the one we find. In other cases, we cannot do this. So if the search space is too large, or if we cannot really find an algorithm that is practical to implement that will give us the solution in an adequate time, then we need to take shortcuts. We saw this with multiple sequence alignment because of the exponential growth of the complexity depending on the number of sequences that we have. And so in that case, we need to take shortcuts to simplify things. And so we need to trade off optimality for efficiency, for being able to find a good solution in a feasible amount of time. So in general, these are called heuristic methods from the Greek word heuristic, for I discover, I find, which has some connotations of luck and chance. So we are taking chances with the optimality of the algorithm by discarding the guarantee that we find the optimal solution, but in return we have more efficient algorithms that we can use in practice. So one approach that we're going to look at is gradient descent. Suppose that we have a cost function or a target function, an objective function with continuous derivatives. For example, one that is widely used is the quadratic error, which is the sum of the squares of the differences between what our model is saying and what the target values should be. So imagine that we have these data here, these data points in blue. Our model is telling us this prediction in red. So for each x value, it predicts a corresponding y value. And we could measure the error as the sum of the quadratic distance between each point and the predictions of our model. So since this is a quadratic function, we can compute the derivative. The derivative is continuous, so we can choose any point here in the space of parameters for our model, and then look at what changes occur that we can use to slide down this function and thus optimize the parameters of our model. So this is one approach that we can use when we have a cost function with continuous derivatives. Examples of this are parameters, fitting parameters of a model. For example, kinetics model. We're going to see this example. It can be used in structural bioinformatics for minimizing the energy of a macromolecular structure. We can compute the derivative of the energy function with respect to moves in the positions of the atoms or in dihedral angles, and then keep adjusting iteratively our structure to minimize the energy. It's used for training artificial neural networks, for example. We're going to talk a bit about those in a few lectures. So there are several applications of gradient descent in bioinformatics. Another approach is the Monte Carlo approach. This name comes from the casinos in Monte Carlo, so it's basically it uses random numbers. It's a stochastic approach to try to either obtain some statistics, solve problems, simulate processes, and so on. A very simple example: suppose we want to compute the integral of this function e raised to the sine of x between some interval. Now, at least as far as I know, there is no analytic solution for this integral here, but we could, for example, if we are doing this between 0 and 8, we can create a rectangle, for example, 8 by 4 units, and then we shoot 10,000 points, which we select 10,000 points at random inside this rectangle, uniformly sampling in both x and y. And for each point we check if it's inside or outside the integral. So it's below or above this function. This is easy to do because we just need to compute the value of the function and check if the point falls below. And we count. For example, in this case, there were 3,677 points inside the integral part here, and the rest was outside. So if we take the proportion of this number to the total amount of points, we have an idea of the fraction of this rectangle, the area of this rectangle, that corresponds to the integral we are computing. And so we multiply this by the area of the rectangle and we get an estimate of the value of the integral. So this would be a very simple example of a Monte Carlo approach to solve a problem which would be difficult to solve in different approaches. So today we're going to look at Monte Carlo as as applied to molecular dynamics. I'm going to talk a bit, not exactly simulating molecular dynamics, but mostly for sampling parameter spaces. There are more complex algorithms. Markov chain Monte Carlo is usually used for this. I'm not going to detail too much about the theory of this, but I'm going to illustrate the applications of Monte Carlo methods in bioinformatics by looking at finding a sample of conformations for a macromolecular structure. We're also going to see more about Monte Carlo methods in the next lecture when we're going to focus on resampling. So the idea there is similar to the example I'm going to give, but it's not quite obtaining an optimal solution, but using Monte Carlo to get statistics on parameters and things like that. We're also going to look at evolutionary computation. The idea is to use populations and selection inspired in biological evolution to improve solutions, and we're going to focus specifically on genetic algorithms, and we're going to look at multiple sequence alignment as an example. Okay, so let's start with gradient descent. Suppose that we have this set of points in red, and we want to try to find the line that best fits these points. So this would be a linear regression problem, but the problem is how do we find the best line? We can have many different possibilities. We need to make some assumptions or think about what would make a line better than others. Now, one thing that if you probably you already did linear regressions, and you know that we find the line that minimizes the quadratic error. So least mean squares is usually what we do, the optimization we do to try to fit these kinds of models. But there is a reason for that. So let's assume that in real life the value of this coordinate y here, so the vertical coordinate, depends on x, some function of x, plus some noise, some random error. And we want to approximate this, our line that we're going to choose, we want it to be as close as possible to this unknown function f that exists somewhere in reality, but we don't know which one it is. But our line will be this function g that depends on the parameters we're going to select. So these parameters theta are the slope of the line and the intercept that determines whether the line is higher or lower. So these are the parameters that we're going to choose in order to draw the best line. And this function here, which results from setting those parameters, we want it to be as close as possible to this unknown f function that relates x with y. And we're going to assume that this error is normally distributed and it only affects y. So you see that y is some function of x plus the random error. And this would be an error that can be above or below. It's a normal distribution. We're going to assume that for the error. So given a training set, this set of points that we have here that we're going to use to adjust our line, we want to try to find the best parameters. And the criteria for finding the best parameters is finding the parameters that maximize the probability of our data under this assumption. That is, in other words, they make the parameters are as likely as possible because they make the data more probable. So this is the maximum likelihood criteria. And the likelihood of the parameters of our model will be the probability of our data, assuming that the parameters are correct. Now, just a brief review of these relations in probabilities. So the joint probability of two variables is the conditional probability of one given the other multiplied by the marginal probability of the other. So we can write the joint probability of having these x values and those y values, so basically having this distribution of points, as the product of the probability of each y given that x and multiplying all those probabilities, and also multiplied by the marginal probabilities of having some point at this value of x or this value of x and so on. So this expression here will give us the probability, this expression will give us the probability of the data if we assume that our model is correct, and this probability is the likelihood of our model. And the likelihood we have the likelihood of this set of parameters that we chose to instantiate our model given this data set that we have here. So this set of points here. Okay, so if we simplify this expression, so we have the likelihood here, it's this set of products. We can take the logarithm so that we can change these multiplications into sums. We can also discard this part because note that our parameters here are the parameters for this function, for the line. And the line is telling us how y depends on x. The line that we are trying to find here, does not tell us anything about the probability of x. So it's independent of that. So this term here, the marginal probability of x, we can ignore because it will not depend on our parameters. It will be a constant. So since we are trying to find the set of parameters that maximize this expression, we can disregard this constant because it will not change the value of the parameters that maximize the expression. And now we know also this conditional probability, because we can write it as a function of our prediction, because we assume that the difference between our prediction and the y value that we're trying to predict will depend on some error that has a normal distribution. So we can use the expression for that normal distribution, and this would be the probability of having that particular shape of the data, so that's a set of values, if we assume that we have these parameters theta for our function that approximates what would be the relation between x and y in reality. So if we simplify this, we use the logarithm to convert into sums, and we remove this part, this e here, because since we are computing the maximum, we're trying to maximize this expression. We can simplify this by simply looking at this expression here. So we try to maximize this part here, which is the power for e, and so basically we are trying to maximize the sum of these quadratic errors between the different the actual values for the points and the predictions in our model. And so maximizing this is the same as minimizing the symmetric, changing the sign that there. So it's that minimization of the quadratic error. And this story is just to understand that one common application of gradient descent is to minimize the quadratic error, and the reason for that is that minimizing the quadratic error gives us the maximum likelihood solution if we assume that all the error is on the target value, the one we're trying to predict. So that is where the noise is, and the input value, the x, the features that we have, do not have error associated. So this is a common assumption when we are trying to fit some model, and that's why minimizing the quadratic error is so common. So the way we're going to do this now is we have this cost function. Sometimes it's written as 1/2, but it makes no difference for the maximum, but it simplifies the derivative when we have these two here coming down as a multiplication. So the 1/2 neutralizes that one, and we have only then the derivative of this part. But the idea is that we're going to minimize this quadratic error by looking at the derivative of this error function at each point that we are considering. So each point is a combination of parameters, and then we are going to move our parameters in order to slide down this error function. So let's go back to this problem here. Suppose we are trying to fit this data with a straight line. So our function depends on these θ1 and θ2 parameters. It's a straight line, and it tells us that the prediction, the predicted value of y is x multiplied by some constant, some parameter that will determine the slope of the line, and then added with another parameter that determines the intercept. So we're going to adjust these parameters in order to minimize the quadratic error. And if we see what this function looks like with respect to the two parameters, θ1 and θ2, it would be something like this. There is some optimal set of values for our parameters, but as we move away from these values, the error increases. So one way of finding this optimal value is to start anywhere, doesn't matter, we choose a point at random. We measure the derivative of the error function there, and we slide down that derivative in order to find some better point in the next iteration. And now we keep doing this, we slide down the derivative again, and we keep moving in steps iteratively until we converge to some point that we can no longer improve. So that would be a local optimum for our loss function. And one good thing about this quadratic error function is that it is convex, and it is easy to converge into the global optimum. So this will allow us to find the best parameters, θ1 and θ2, so the best slope and the best intercept for this line. Okay, this is not a very good model, a linear model, but we're going to see a bit more about machine learning and supervised learning, regression, and so on in a few lectures. And we're going to see another example of this approach using gradient descent. But this gives us an idea of how gradient descent works. So basically, we use the derivatives of the cost function in order to find the minimum of that cost function. So let's see a practical example from biochemistry. I think most of you are familiarized with the Michaelis-Menten Kinetics. This is a very basic model where we know that the initial velocity of an enzyme reaction will depend on the concentration of the substrate, and it has some saturation that is determined by this Km value. This is the concentration of the substrate at which the initial velocity is 1/2 of the maximum possible, and the maximum possible velocity for that enzyme. So this would be the relation between the concentration of the substrate and the initial velocity measured of the reaction, and it depends on these two parameters, the maximum velocity and the concentration at which the initial velocity is 1/2 of the maximum. Now, suppose that we did these experiments, we tried different concentrations of substrate, and for each concentration we measured the initial velocity. So this gives us a set of data points. And if you don't want to use programming and computers and so on, there is a way to solve this by linearizing using logarithms and so on, and then doing a linear regression. But if we do the regression directly, we can consider explicitly that the error is in the estimate of the initial velocity. There is no error there in the substrate measures, because we can consider that we quantified the substrate carefully, and so we can do a least mean squares regression directly on this expression and get the curve that best fits the data that we have. So a general recipe for applying this approach would be to find a reasonable cost function. That sum of squares is very common, but in theory for gradient descent any function that has a continuous derivative could be used. And then we're going to slide down that loss function to find the minimum of the cost or the error for adjusting the parameters of our model. So first we can write the cost function here. We need to do it in this way. We're going to receive a tuple with the values to try for this particular point, the values to evaluate. And so we have here the first parameter, which it would be the Vmax there, and then the second parameter is Km. So we're going to receive that as a tuple with two parameters. And we also want to have the concentrations of substrate and the initial velocity. So this would be the experimental data here, one vector with these values and another vector with these values here. So we're going to compute the predictions, that is for this set of concentrations. We're going to use the two parameters, the Vmax and Km, to predict what would be the initial velocities for each of these values if these parameters were correct. And now we're going to compute the sum of the squares of the errors by using the actual measured initial velocities minus the predicted velocities squared, and then we sum everything. So this would be the cost that we have here. Note that you should test your functions before continuing with the code and combining them with other functions. I'm going to omit that, but that would be an important step when you're writing code. So now we're going to use from the SciPy library, there is this optimize module, and we're going to use the minimize function. So what the minimize function does is going to try to slide down the error function in order to find the optimum value. Now, these functions actually work even without the derivatives if you have a very simple problem, but if you have more complex problem, you need also to provide the derivatives for the loss function so that they can solve this problem of figuring out in which direction to move the values in order to optimize this target function. So what we need to do in this case is just to give the cost function that we want to minimize, and this function must be of this format. It's going to receive all the parameters in a tuple, and it may have additional arguments that we specify here. So these are the additional arguments that we took from the data. The concentrations of substrate are in the first row of our matrix, and the velocities are in the second row. So this is our matrix here with the concentrations of substrate and the velocities, the initial velocities we measured. So now when we call this function, it's going to try different values starting from some point that we give it. So you can do this, for example, start with 1 in Vmax and 1 in Km. And we can also include boundaries for the values it's going to try. So in this case, we do not want negative values either for Vmax or Km, because that would not have a physical meaning. And so we set the bounds for our two parameters as being between 0 and none. None means there is no upper bound. It can increase as much as necessary. So when we call minimize, we get a result object that has this x attribute, which contains the set of values that the minimize function found to minimize the cost function we provide here. So since we are using Vmax as the first parameter and Km as the second parameter here, we can now unpack this result into Vmax and Km and just return the values. Now, if we want to see the result, this is often useful. We're going to create here an array, a vector of x values that has 200 points, and it's uniformly distributed between 0 and the maximum concentration of substrate we used multiplied by 1.1. So this will give us a set of 200 points here that goes a bit beyond the end of our data and allows us then to draw this smooth curve, because note that when plotting, the plot library will join consecutive points by straight lines, and if we don't have a large number of points, we cannot represent curves in a smooth way. So we're going to do that. We're going to take 200 points. We're going to compute the initial velocity if we would use all of these points as the substrate concentration. So this is the Vmax parameter multiplied by all of this vector and then divided by Km multiplied by the vector. So this gives us a vector of y values for all our predictions, this red line drawn here. And this we can now plot in this way. We're going to create a figure. So you import this module, pyplot, from the matplotlib library. This is usually the nickname given to this plotting library. And what we do is we can create a figure. This is useful if you want to give it a size to specify the aspect ratio and so on. So the fig size is first width and then height of the figure. And now we can draw the line by giving the x and y coordinates, and then this format string specifies that we want a line and we want the color to be red. This is the R there. We can also specify the width of the line. It can be narrower or wider as you wish. And then we're going to also plot our data. So our data is this matrix here. This would be the x coordinates, and these would be the y coordinates, the initial velocities measured. So we're going to plot that, but we're going to plot it with crosses instead of joining with the line, and in blue. We can also choose the size of the marker, the width of the marker, and so on. So you have these blue crosses here to indicate where the original data is. And if you want, you can save to a file. So this would be this function to plot would receive the name of a file, some file.png, for example, and then it would save the image there. Alternative, if you want just to see the image on Spider, you can omit these. So you can do it like this. When Spider detects that there is a plotting object in memory after your program ends, it will show you on the plot window. So if you just plot and let your program finish, you can see the plot in the plot window. So this would be the result in this case, and we would have the Vmax and the Km values for this particular data given our assumptions that we only have an error on the estimates of the initial velocity, and we obtain the best model here. Note that this is a very simple example. If you have more complex examples, you need to actually give the derivative and so on. Otherwise, you will probably not get a good result. So this is just to illustrate the idea of gradient descent, although in this particular case, it's so simple that we do not need to use gradient descent. The minimize function can try to infer the derivatives numerically from the points that it's estimating. Okay, so in general, gradient descent can be efficient if we have convex functions and they are continuously differentiable. So that means we can follow the derivatives and find global optima. It can give us the optimal solution in that case. For example, these simple quadratic error functions with few parameters usually work very well, but it cannot be used if the function is not continuously differentiable. So if you try to do that, this kind of approach will give you the incorrect results because there are discontinuities in the derivatives, and these minimization methods are trying to use the derivative, whether inferred numerically or given explicitly, in order to determine which would be the next step. So if the derivative has discontinuities, the solution can be very wrong. And even if it's continuously differentiable, there may be local optima, and as we are sliding down and stopping when we find a point which we cannot improve, we can get stuck in some local optima that are very worse than the global optimum we were trying to find. Okay, another approach is, instead of trying to follow the derivatives, we shoot at random and try to get some results from there. For example, this is very used for thermodynamic studies of proteins because free energy will depend not only on the energy of each individual configuration, so the enthalpy, but also on entropy considerations, on the probability of finding some family of conformations. And this can be estimated by sampling conformational space. So the general strategy for this particular application of Monte Carlo methods would be to do some random moves. If you want to optimize the structure, you can just select the random moves that improve, for example, by minimizing the energy or something like that. If you want to obtain a population of likely conformations, you can use some probabilistic criteria for accepting or rejecting each alternative. So what we need is basically a set of legal moves, what changes we are allowed to make. So in this case of protein conformations, we are going to use the dihedral angles in the backbone and the side chain. There is an E missing there. There's a typo. And we also need an energy function so that we can estimate the energy of each conformation. And we need some criteria for accepting or rejecting each of the random conformations we are going to obtain. So the move set in this case for protein structure and for thermodynamics or for optimizing protein structure is to randomly change dihedral angles. And a dihedral angle in general is the angle formed between two planes. In the case of molecules, each plane corresponds to 3 atoms, and the dihedral angle is a rotation around a bond between 2 atoms. So if we have 3 atoms, we can compute a vector that is perpendicular to the plane formed by these 3 atoms by considering 2 vectors, one which is atom A, the coordinates of atom A minus the coordinates of atom C, and the other B minus C, the coordinates of B minus C, and we can take the cross product of these 2 vectors and obtain one that is perpendicular to both. So this way it's a vector that is perpendicular to the bond between A and C and B and C, with C being in the middle of the two of these 3, and so it's perpendicular to the plane. This is easier to see graphically, so let's consider this stretch of a protein chain. We have a valine residue here. This is the nitrogen of the valine, the alpha carbon, the carbon of the carboxylic terminal here, and then this bonds to the next residue, and this here is bonded to the previous residue. So let's consider these 3 atoms here first, centered on the nitrogen. So we have these 2 bonds here, and we can consider a plane formed by these 3 atoms. So if we take these 2 vectors which correspond to these bonds and compute the cross product, we obtain a perpendicular vector here, centered on the nitrogen. We can do the same thing for these 3 atoms here. So the nitrogen, the alpha carbon, and the carbon of the carboxylic atom, of the carboxylic group, sorry. And we have here a vector that is perpendicular to this plane. So this vector indicates the orientation of this plane here, and this vector in red indicates the orientation of this plane. As we rotate around this bond, the 2 vectors will change orientation relative to one another. So you see we are rotating around the bond between the nitrogen and the alpha carbon. And if we look at this in the direction of this bond here, we can see the angle between the 2 vectors changing. So this is the dihedral angle that we can use to represent rotation around a bond between 2 atoms. So now what we can do is from a structure of a protein, we can do random perturbations to dihedral angles by small amount, and this will change the conformation of the protein. We can do this either in the backbone or in the side chains, and so completely change that conformation. And now this may lead to atoms being very close together, high energy conformations and so on. But now we can check that by looking at the energy function. We can measure the energy of, or estimate the energy of that conformation by looking at different aspects, different components of the energy. So these parameters and the different components that we are using are called the force field in structural bioinformatics, in this kind of problems, and they are composed of different aspects. For example, the Lennard-Jones potential is composed of the repulsion and the van der Waals attraction. Repulsion when we're trying to overlap 2 electron clouds too much. When we push 2 atoms together, this makes the potential energy increase a lot. And the van der Waals attraction, because of what happens when these electron clouds polarize, and they may at some distances even slightly attract one another. So we can, depending on the distance between 2 atoms, we can compute the contribution to the energy of those electron clouds. We also can consider the Coulomb potential, because the atoms may have partial charges depending on the distribution of the electrons, and so we have to consider the attraction or repulsion between 2 atoms depending on the charges that they have. And then there are other factors here we can consider, distortions in the bonds. If we can allow changes in bond length and bond angles, we may need to compute those in the force field. If we're only looking at dihedral angles, we can also take into account the torsion and other forces around those angles, using functions that would estimate the energy of different dihedral angles. So taking these together, we can create a potential function, something that tells us the energy of each conformation, depending on the charges of the atoms, the typical dihedral angles, and so on. And we could evaluate the energy for each conformation after inserting those perturbations. And now we decide whether or not we're going to accept this new change. So one common criterion, the Metropolis Criterion, will give us a probability of 1 of accepting the change if the energy decreased, if we lowered the energy, or a probability of E raised to minus the change in energy divided by the temperature and a constant. But this basically means that the higher the temperature, the more probable we are accepting one change that increased the energy of the system. But if there is a decrease, we accept it automatically. So we can use this, for example, for simulated annealing by gradually lowering the temperature and changing the conformation iteratively, so we can explore different conformations, leave local optima, and then start to converge to some optimal solution as we lower the temperature. So this could be if we want to optimize, for example, minimize the energy of the conformation of our protein, or we can even just use this to generate a population of conformations and to get an idea of the distribution of possible conformations. So this can be used to estimate entropy of different parts of the protein and so on. There are many complex details in real applications. So molecular dynamics is a complex field. Also, these Monte Carlo Markov chain Monte Carlo methods that use these chains of perturbations of the conformation are fairly complex. But the general idea is that we're going to do random perturbations in the conformation of that structure, evaluate the energy, and then decide whether to accept or reject. If it's simply an optimization problem, we generally want to accept anything that improves the energy or lowers the energy, and we only accept with some probability things that increase the energy, changes that increase the energy, because that will allow us to escape local optima and not get stuck in some conformation from which we cannot move just by lowering the energy. Another approach is to use randomness, but inspired in biology. We're going to look at the populations, evaluate these populations, and then select the best candidates for the next generation. So basically, we're going to solve optimization problems by using the same kind of mechanisms that biology uses in evolution. There are many different types of evolutionary computation algorithms. This includes modeling interactions, like modeling the individual agents, artificial life, algorithms that mimic the way ants behave, so to try to improve search of different solutions and so on. We're going to focus on genetic programming, on genetic algorithms. Genetic programming is an application of genetic algorithms to actually try to write computer programs, but we're going to focus on genetic algorithms which have a basic structure like this. We need a representation of our solutions. So we have the DNA of our individual, so to speak, and then we need to map this to the phenotype, to the actual individual. For example, in protein-protein docking, we need to figure out what is the relative position of one protein relative to the other that maximizes the contact between the two. We're going to assume that the two proteins at that scale are rigid, so we need only to rotate and translate one of them relative to the other. This gives us 6 degrees of freedom for the rotation and translation, so the DNA can be a series of 6 numbers, and the phenotype would be the actual placement of the protein in that position, in that orientation and that translation. If we are doing, for example, ligand protein docking, where we have one small molecule that can be very flexible, we can consider 6 degrees of freedom to decide the position and orientation of the ligand relative to the protein, but then more degrees of freedom for all the dihedral angles in the ligand we may want to be able to move in order to change conformation of the ligand. So this would be a sequence of numbers that would be translated into the ligand with some specific conformation at that particular position. In some cases, the genotype and phenotype can be exactly the same. For example, in a multiple sequence alignment, we can use the actual alignment as the genes that we are going to recombine and mutate and so on. Another thing that we need is to evaluate fitness and select candidates for breeding for the next generation. So we need some scoring function that tells us which are better, which are worth, but this, of course, we also always have when we are dealing with an optimization problem. We need the function to optimize. And then we need these operators. Crossover operators are those that allow us to mix together characteristics of the parents when we create a new generation, the children for each generation. And mutation operators allow us to introduce random changes, so to not lose diversity because of selection. And we can keep introducing changes to the representations of our individuals. Okay, let's look at a concrete example of this so that's easier to understand. And actually, if everything goes according to plan, this will be your second assignment to implement multiple sequence alignment with genetic algorithms. So based on this paper here, I'm going to describe briefly what the authors did. And the initial population is a set of random multiple sequence alignments, which are obtained by aligning all the sequences by putting between 0 to 50 gaps at the beginning of each sequence at random. So you can imagine that you have a number of gaps, then the sequence, and then the other sequence on the second line will have a different number of gaps at the beginning. And so all the sequences will be a bit scrambled in the multiple sequence alignment by having a random number of gaps in the beginning. Then the end will be filled so that we have a rectangular multiple sequence alignment with all of those sequences with the same length. And now we're going to score each alignment, and the score will be used to obtain the fitness for the next generation. There are many different schemes for scoring the alignments. We could count the identities, we could use the sum of pairs, the weighted sum of pairs. We saw some of those in previous lectures. But once we have scored the alignment, we can now assign a fitness value for each of our individuals. So basically, if the score is something that we want to maximize, we can use the score as the fitness value. If the score is something that we want to minimize because it's a cost, then we can use, for example, 1 over the score as the fitness value. But the idea is that fitness will tell us how probable it is that this individual will be selected for the next generation, and the higher the fitness, the higher the probability. Now, in this particular case, each generation was created by copying the top 50% individuals of the previous generation. So the top half of the population was copied into the next generation, and then the other half was filled in by selecting individuals at random and crossing them with each other. So each pair of individuals, we crossed the characteristics of those individuals. Now, this selection is at random, but it's not uniformly distributed. So individuals with higher fitness are chosen with larger probability. And this will gives us a balance between exploration of different combinations and exploitation, or greedily trying to improve the fitness of the population. Now, this the way to do this crossover, to mix two individuals, was using a point crossover. So in this case, for this particular application, the authors chose given two parents. So suppose that this is parent A and parent B. These are two multiple sequence alignment. We choose a line here breaking parent A into two halves, a vertical line splitting between these columns. And then for parent B, we split at the same position. For example, between this T and this C, it would be the split there. Between this T and this G, we would have to put the split there, and so on. So that now we can combine the two. But note that since the alignments are different, splitting at a straight line on this one will result on a ragged line probably on the other, but we need to split on the same points on all the sequences. So that now we can mix these in the next generation by taking one block from one of the parents and another block from the other, and then adding the gaps as needed to create a new multiple sequence alignment from these blocks. This is quite disruptive because it changes a lot the alignment. So another crossover operator that the authors used was to find columns that were the same between the two. For example, these C alignments here were the same between the two parents, and then exchange the blocks that are between the identical columns. So identical columns are preserved in the offspring, and only the parts that are between are scrambled between the two parents. So these crossover, the crossover operations are those that allow us to mix characteristics from the two parents in the offspring. And then there are mutation operations, for example, looking at the guide tree and choosing a point in the guide tree to insert a gap somewhere. And so all of the sequences that are affected there following the guide tree of the multiple sequence alignment would be affected by that gap. So this requires computing the guide tree. It can be computed directly from the multiple sequence alignment by looking at all the pairs and doing what we saw before. So this would be a phylogenetic tree, but this would be one way of doing the mutation, or we can just insert gaps somewhere randomly or something like that. So there are many different ways of mutating the individuals that we are considering. So the general idea here is to use a population. In this case, it's a population of multiple sequence alignments. We need to score the individuals. We can score each multiple sequence alignment and then use those scores to select the candidates for the next generation. Then we need crossover and mutation operators so that we can combine different parts of the parents and insert diversity into the population with these mutations. And now we're going to repeat this by going from generation to generation until we have solutions that we find adequate. So to sum up, we saw previously that there are optimization problems where we can guarantee an optimal solution, for example, aligning two sequences, but then there are more demanding problems that require taking shortcuts, heuristics. And now here we saw three different approaches, sliding down the derivatives to try to reach as fast as possible the optimum. This is good if we have an objective function for which the derivatives are well behaved. If not, then sliding down the derivatives will mislead us. And it also, even in that case, it may get stuck in local optima. So it's best for convex functions that have an easily accessible global optimum. Another approach is to use randomness to try to improve things or to sample populations. We're going to see in the next lecture the application of these methods for resampling and for taking statistics, but you can use this for optimization too by using some criteria for whether or not to retain another solution. So this randomness can be used to escape local optima, not get stuck, and explore a bit more of the space before we converge to some optimum. And we can use evolution. This is inspired in nature. We can use populations of candidate solutions, select the best at each generation, cross the attributes of the parents, mutate, and keep repeating that generation after generation until we have good results.